Okay, so it is recording. Okay, official welcome to everybody. We have with us Dr. Lee Miller and Dr. Doug Richmond. Dr. Miller is our turf pathologist stationed on campus. Dr. Richmond, turf insect, is that how your title there, Doug? I want to get your title right.
Turf entomologist, sure.
Turf entomologist there. I knew I was goofing up a little bit there. So again, please answer that demographic poll if you don't mind. We'll get that information. There is a lot of information to be covered today. So any questions that come up, throw those in the chat box and we'll get those as best as we can. Like we said, we've got a good crew today. So if you don't mind keeping your microphones muted, we'd appreciate that. And with that, it is now 12:02. Dr. Miller, the floor is yours.
All right, let me switch over and share my screen here and we'll get started. Thank you, Jeff, for this invitation to speak at this hot topics. Good morning, everyone. And I'm not wrong. It is morning here. I'm in Madison, Wisconsin. I'm attending a conference. It's again, I'm about to head back to Indiana here in a few minutes. Let me get that poll off my screen. In a few minutes, but again, I'm really appreciative of this chance to speak with you and to meet some of you. So I think I've met a lot of the extension educators at our professional development conference, but I signed on a couple years ago. So I moved from Mizzou to Purdue in 2022. That was the campaign button that I created for my family. So it's been a pleasure to work here, and I've got great colleagues, and you'll hear from Doug Richmond, one of my favorite, and I'll say that to your face, Doug, one of my favorite colleagues since moving here. I'm a turfgrass pathologist, so I've had to explain this to my mother. I'm a coroner for grass, the legal kind. I tell folks why their grass died, and I tell them how to fix it. If the plant is over 4 inches, I'm out. So I work a lot with golf courses, as you can imagine. They mow grass to a tenth of an inch and right on the edge of its life. So I work a lot on golf putting greens, but I do also work a lot with homeowners, and I'll highlight that towards the end of the presentation. So I do research, so I'm not so morbid all the time. And I have 2 new graduate students that just started, well, actually last summer and in the fall, Mariah Cashbaugh and Justice Ruana. Very different graduate students. I'm glad to have both of them. There's a lot of diversity of ideas from both of them, and actually even their personalities, which is kind of fun. So I'll talk about each one of their projects as we go forward, and I'll start with Mariah. I also will say that it's nice to be young again. I try not to call any of the students kids, but then when I don't know if most of you probably don't know what this is, but this is a -80 freezer. And this is how I opened it up the other day. So I haven't run a Spanish Inquisition and figured out who did it. But I'm just going to leave it there because I think it's kind of fun. So we do have a little fun in science and research. So anyway, just kind of humorous. So Mariah's project deals with plant pathogen detection and percolated groundwater. So most of you might not know, but golf course putting greens are very artificial systems. They're actually, most of them are built to this USGA putting green profile where we have a sand base. It's normally an 80-20, so 80% sand, 20% organic matter, or sometimes calcined clay or profile. That is 12 inches over a pea gravel layer. And then out of the bottom comes this drainage pipe. So this is really one of the few agronomic systems where we might be able to get to some of the percolated groundwater. And we're wondering if we can actually detect some of our plant pathogens in this percolated groundwater. So our first job is, I can't say that at the bottom there, that is also called a sub-air system. There's another one called PA, or precision air. And some golf courses, some of the higher ends, also have basically a big shop vac that they can hook up to their drainage system and actually suck out the water, or sometimes they blow in cooler air. So a very different system than you probably would see in almost any other agronomic or horticultural crop. So our job first is to figure out if we can detect plant pathogens in the percolated groundwater. The idea is, can we actually monitor inoculum levels as they go throughout the season? And then if we can detect it, that means that with some of our molecular analysis we can do now, we can also quantify it. So then we can actually start tracking this throughout the season. Obviously, one of our first targets is going to be soil-borne pathogens, so things like summer patch or Pythium root rot. But also we can also monitor biocontrols to see if they're actually accumulating in the system. And actually, one of the problems with organic disease control is we often try to use a biocontrol agent, and it's basically like trying to throw a baby out in the middle of a bunch of lions, because it just can't. The habitat is so rich there that microbial diversity is there and is ingrained. They're already kind of home, and then you've got another invader that's coming in. So we might be able to track that through this percolated groundwater. And then also we've got some threshold models, and maybe we can actually correlate and refresh some of these threshold models that we have that are based on the environment, and actually do that and correlate it directly to the inoculum levels. So this is our first crude crack at it. We're doing some work in the greenhouse, and we've gotten better than this, but this is actually some cut up gloves, and we're just trying to inoculate on the top and run some things down. We're also doing some on-course sampling. So we have found a lot about putting green drainage that I actually didn't know. Most superintendents actually don't know where their drainage outlets are from the putting greens. So that's another subject to talk with them about altogether. So this is down in a hole. This is actually one of the good ones. This is one of the more challenging ones. I didn't realize I was going to have to buy rappelling gear for some of these drainage outlets. And then we also, are getting down and getting in and putting our head in holes, quite honestly, to try and collect some of this groundwater. So that's been a very fun part of Mariah's project, and we look forward to moving forward with it as we go through. Also want to describe Justice's project. So we're looking at trying to interseed resistant bentgrass cultivars into golf course fairways. And basically there's most of the fairways that we have in Indiana are a susceptible variety. And we're looking to try and reduce the amount of fungicides that are applied on bentgrass fairways, which are larger acreage. We're normally looking at about 30 acres per golf course. And there's 411 golf courses per just in Indiana alone. So if you start thinking about that and the math of this is actually the largest area that's probably, particularly in turf, that constantly gets fungicide exposure. So we're trying to integrate host resistance, which we know is one of the first keys of our IBM practices. So we know that a lot of when I was at Missouri, I maintained all of our NTEP trials. And this is basically dollar spot AUDPC, or area under the disease progress curve. And this is from the 2015 bentgrass test. And you can see the stark differences in the amount of dollar spot that we're getting in each one of these cultivars. And if you go up to 2017, you can see, while some of the levels change somewhat, you can see what the difference is from a very highly susceptible cultivar down to one that we would consider to be resistant. And then this also, Cole Thompson from the USGA, now at the USGA, also did some work in this area. You can see the stark levels of difference that we can get. So the question becomes, well, why isn't everybody doing this? We've got decades of work by breeders to develop these cultivars and to understand the pathosystem, to understand the genetics. Why isn't everybody just willy-nilly doing it? If you look at the potential just to reduce one fungicide application, if we did this on all 411 golf courses in Indiana, one fungicide application would be $2.5 million. So we're actually looking to do even more than that. Some of the drawbacks, though, is, obviously one of the ways to do this, and the only way that's really thought of, is to do a hard reset. And that's basically to apply glyphosate over all of the fairways and to start completely over. Obviously, you have, of course, where that has lost revenue and basically just unsatisfied customers at that point. There is also a weed called annual bluegrass, or Poa annua. If you listen to some of the golf commentators, they call it Poe, that we can introduce by taking the bentgrass out. So once you remove that cover, Poa annua really likes to come in. So we have that aspect. And then last but not least, there's an unknown agronomic quality of some of these new cultivars. And basically, I had a superintendent come to me and go, listen, if I were to try to do this on my course and it didn't work, I would lose my job. And there's no two ways about it. So that is another aspect of this. So we're thinking maybe we can use some different techniques to interseed these cultivars and kind of do it lightly. So you still have play, you still have everything else that's going on, but maybe over time you can interseed these cultivars and build those levels of host resistance. So we have a 3-year multi-state field trial that we initiated last July. We're interseeding 3 different resistant cultivars into a susceptible bentgrass cultivar background. And I also will say that there's POA in all of these sites as well. The cultivars were split seeded in 2 directions for 2 total pounds per 1,000 square feet. And we use this specialized device called a Varieto Turf Fix. And I have to give a shout out to the person that's on that. That's Sam Stimmel from AquaAid. He actually drove the unit from Pennsylvania to us on the day after July 4th, so July 5th. He didn't like the way it was set up. So he actually drove to Cincinnati to get it serviced, came back the next day, interseeded with us, went up to Wisconsin and interseeded them the next day. So we do have some pretty incredible industry partners. And quite frankly, it's a pleasure to work in the turfgrass industry. We have that much cooperation and really interest in the research that we're doing. So this is what the plots kind of look like. This would be one replication. We've got the 3 different cultivars there that are in the different colors. And then we also added, well, glyphosate is the hard reset. That would be the entire course closure, or at least the some of the holes. And then we added different herbicides and PGRs in there to see if we could knock back the POA. We do have a lot of POA contamination as soon as you disturb a site. So one of the reasons we didn't start this in a year earlier is that one of my esteemed colleagues said, you're getting nothing but POA if you do this. So we decided to add some herbicides and PGRs in there to try and knock that back and to see what's going on. The other aspect is, if you notice the Fs there, we are going to use a threshold. So we're going to count to 5. And then we're going to apply a contact fungicide. And the idea there is we're going to have one we're going to let go to nature. And then the other half of the plot, we're going to see exactly kind of how many licks does it take to get to the center of a Tootsie Roll Pop? How far have we moved along the host resistance to see if we can actually limit the amount of fungicides that we're using? So this was, if you noticed earlier, this was financed by the USGA, the United States Golf Association, one of our major funding partners. And this is a multi-state. So I talked about University of Wisconsin. That's one of the reasons why I'm up here, to see their trial and actually to bring back some samples. We're also doing this at Penn State University, and that is all being led here at Purdue. You also notice the USDA ARS there, and we've got a partner that has designed molecular markers for each one of the cultivars, and we're sending in clipping samples. So he's got our background, what we started with, and then he's actually going to be able to molecularly analyze and tell us what the population distribution is and how far we've kicked the can down the road to integrate some of these resistant cultivars. Interestingly, he's also going to be with digital droplet PCR looking at dollar spots. So he's going to be able to determine how much dollar spot is in each one of the plots, and then also how much POA is in the plots. So we're not just doing this from a visual level, we're also molecularly analyzing the differences among these populations. So I'm very excited about this study. We've also got a parallel greenhouse study that we're doing where we seeded these in at the same time. So we kind of cycled up. So resistant cultivars, we got it at 0, so just the susceptible, and then 10%, 25%, 50%, 75, 90, and 100. And the idea there is, how are we going to be reducing this in the field? So when we get these percentages in the field from the molecular analysis, what do they mean? How are we actually decreasing the amount of dollar spot? And from some of our data that we've processed already, we're kind of seeing that real decrease come at about that 50-50 level, as far as the size of that lesion and the size of the spot that we get. So last but not least, on the research front, I also am collaborating with several other entities. So this is ag and biological engineering. I had one of the new assistant professors. So one of the really cool things that Purdue does is they take all the assistant professors and they put us on a bus during the summer, and they take us to Southern Indiana, Central Indiana, and Northern Indiana. And doing that, we get a lot of partnerships and a lot of chances for collaborations. So this is with Kurt Ristroff as the PI. Louisa is the graduate student. I didn't make her smile. She actually just naturally smiles when she's doing all this work. I like to see that enthusiasm. But Dr. Ristroff said, I think I've got a formulation that can move the fungicide not only up in the plant, which we're used to, which is acropetal movement, which is what almost all of our fungicides do, but we also can move it down in the plant. And quite frankly, I don't believe him. And that's why you do research with someone. It helps to be a skeptic. So we're just embarking on this research and seeing if they can mess with or at least design fungicide formulations to actually move down in the plant, which would have huge implications for our disease management with fungicides. One of the reasons for that is because all the fungicides move up in the plant. Turfgrass is the most pruned crop on the planet. So we actually mow off our fungicides that we put on. So if we can actually have it more internalized, perhaps we can have longer residual. Now let me get to the other part of my job, which is the extension part. And I want to particularly focus on the homeowner portion. I'm not going to talk about golf courses anymore. But the homeowner portion, a lot of times I'm seeing that homeowners, and I know there's some master gardeners on here as well, homeowners make it very, very difficult to manage turfgrass. They think that it's a very difficult thing, whereas really it needs to be fed, just like me. You don't see my profile, but I eat. And sometimes it needs water. Most of the time it doesn't actually. Most lawns are not irrigated, and it needs to be mowed at the correct height. And really a lot of when we do see problems, over half of what I diagnose is what I call abiotic disorders. It has no disease that is causing the symptoms. Most of the time it's something that's a human error are along those two things. And one of the bigger issues that I've noticed throughout my career is most homeowners don't realize that they are dealing with a very substandard growing condition to start with. So particularly if you have a newer home construction, this is what you're left with. A lot of times the topsoil is scraped off and you're left with the gray and brown subsoil. And quite frankly, gray and brown to me mean dead. So we often think of, oh, well, we're in one of the best soils in the world for agricultural production, but that's not really the case when it comes to home construction. So take with you that this not only goes to lawns and to turfgrass. I actually think it's a miracle you can grow turfgrass on this starting soil, but also to everything else that you put in the landscape. And this is really an underlying reason. So, when you're out and if you're working with homeowners or you're working with your clients, take a screwdriver out and try and push it in the ground. Because if you can't push a screwdriver in the ground, guess what else can't go in the ground? Roots. So just keep that in mind. This year we had everything, all of the biology of the plants. Our diseases started 2 to 3 weeks earlier. So even since I've been here since 22, dollar spot started 3 weeks earlier, May 1st. This is it on creeping bentgrass. We haven't really seen it on Kentucky bluegrass lawns yet. And then the disease on, I'm not sure on your right or left, but started May 21st. That's brown patch lesions on tall fescue. So both of those started 2 or 3 weeks early. So part of my summer I spent down in Louisville. And this is the 9th hole at Valhalla Country Club. So I spent a week there volunteering. I forgot how much time it does take. But one of the aspects I want to point out there is look at what I'm holding there, a blower. And I'm going to get back to that in a little bit. But that is one of the most least used implements in the lawn and landscape sector, particularly by homeowners. But I will tell you that it is one of the most used implements in golf courses and in high amenity. So I'll talk about why that is. I want to start with one of the first cases here. I just have two really brief case studies. This one was submitted actually just last week. And this is a homeowner that complains that every year, basically his lawn starts declining this time of year, and that's basically he wants to have the best lawn in the neighborhood, and he just can't keep things going. So he's actually got a mix of Kentucky bluegrass and tall fescue. He actually has a lot of tall fescue in this fairly good looking lawn from here, but he's starting to get these spots. And this is a disease that I don't normally see on tall fescue, but this is bipolaris leaf spot or leaf spot melting out. And it's actually unlike any that I've ever seen. I've never seen this amount on tall fescue. This can be attributed to how wet we were in April and May. Particularly in Indianapolis in April, I think the number is somewhere around 7.7 inches, or nearly twice the amount of rainfall that we normally would get. In May was also a couple inches over normal as well. So we had a very wet April and May, and basically that is causing these outbreaks. And I think he's seeing this year after year. Now, what's unique about this particular situation is that this homeowner is extremely aggressive in his maintenance practices, like nobody I've ever seen. So actually, I responded, and this is the longest report I think I've ever written and the most detailed. The homeowner is a lawyer, so take that as you will. But what I want to point out here is under the fungicide. So he was applying fungicides on 2-week intervals, starting on March 7th, and he still was getting the disease. So I'm still a little confused why, but what I want to point out is the first 3 of those where they're actually wasting money. So there is no need on home lawns to apply fungicides on 2-week intervals. Most home lawns don't get a fungicide application at all. So there is an aspect of, I'm not sure about how much fertilizer he was putting down. He doesn't have in-ground irrigation. He says he waters by hand, which makes me wonder if he was watering by hand, which he didn't need to in April and May. So there's a lot going on in these kind of situations, and I think it's important that that you have these questions that you're armed with about irrigation, about fertilization, and then if they're applying this much fungicide, how are they doing it? So this is not the only one. This is one of 2 that I've actually had where they're applying on 2-week intervals. And that's really concerning to me. There's really no need to apply. Like I said, there's no need to apply fungicides on 2 weeks in intervals unless you're doing something else extremely wrong. Mowing too low, fertilizing extremely high, or in particular in-ground irrigation that is put on that is basically set it and forget it. So you'll notice that's the biggest font that I can put on that knot. We don't need 2-week intervals. The other thing that's concerning is that homeowners are reaching out to fungicide dealers. So they're not going to Home Depot and Lowe's anymore. They can buy these things on Amazon, or there's a number of other purveyors, or they're actually going to a dealer that a professional would go to. And I've experienced this both that last one and then the other one. Some are applying off label. There's a number of fungicides that are not labeled for residential, and they're applying those on sites that they really shouldn't be. Also, there's a lack of equipment and expertise, particularly for any of the sprayable formulations. You're not going to be able to get a good distribution if you have a pump up sprayer. It's not going to happen, and I think that's what's going on in a lot of these cases. Last but not least, it's really simple. Tall fescue is one of the strobilurins for brown patch control, azoxystrobin. Most of the time, you don't even really have to do that. And then Kentucky bluegrass, if you do have dollar spot or some other or leaf spots that come in, you might need to add in a DMI fungicide, something like propiconazole or myclobutanol or one of those. So I think we're moving away from the KISS model to keep it simple. And I think I'm seeing some homeowners that are really over maintaining the lawn and putting on too much chemical input. Along with that, I would like to launch kind of an initiative, and I'll call it Bibbity. And one of my frustrations is that most homeowners don't understand a blower and how to use it. So although you can't see it here, this is after fertilization, and this is all the fertilizer prills that ended up on the sidewalk or the driveways or the roads. All of that is going into our road, all into our sewage, all into the road, all through the gutter system and into our streams and waterways. So I would like to try and get a campaign going, and I call it Bibbity. And I don't know if I'm going to get sued by Disney, but basically Bibbity Boppity Boo, right? That's not how you spell it. But basically that stands for blow it back in the yard. And the blower is one of the most environmentally responsible pieces of equipment that you can use. And whenever you mow, obviously clippings, the homeowners association is not going to like that. But also anytime you're using a spreader, all of that needs to go back on the lawn. We've proven that if fertilizers and other amendments go onto the lawn, they're for the most part taken up and locked up, and they're not going to get into our waterways. So that's one of the messages that I want to try to get out, hopefully through our Turfgrass extension educator system. And then we often hear about mowing height, right? So higher mowing heights and I'm not a weed scientist, that is Aaron Patton, but we know that we want to try to make the turf the trees of our lawn. And the way to do that is to set the mowing height all the way up at as high as you can. But one of the things we forget is that after we mow, sometimes we leave clippings. And those clippings act like basically a big tree. They shade out and sometimes they mat down. Underneath, we're going to get high humidity. We're going to get diseases under there. And this is just an example of where we have clumps of clippings that are left on top. So I know I look like a maniac to my neighbors, but after I mow, I actually walk around not only just on the outside, but also in the lawn to disperse those clippings. It's not that hard to do. And that really, I think, is one of the leading factors of some of these diseases and some of our lawns that are declining. So to learn more about diseases as they occur throughout the season, I author a number of disease reports. Actually, I have one due this week, and my head due. But you can find it at turf.purdue.edu. They're in the category disease reports. Also, they're right there on the front. Basically, I go through the weather since that really drives everything that we deal with as far as diseases and also turfgrass health. And then I'll get into one section that's lawns. I'll talk about, in this particular one, seed heads or lawn specific problems. And then I'll go on to golf courses towards the end. And I normally pick something that I kind of center on. I disperse these through my Twitter account. So this is at Purdue Turf Path. If you want to follow me, you'll get an update of when I release some of these. And also if there's something really immediate that I feel like I need to get out. We also have our Turf and Landscape Field Day on July 16th. I just wanted to put out an advertisement for that. And with that, this is my contact information. Again, Jeff, thank you for the invite. And I'll answer any questions if there are some.
And threw up some of the links to your pathology information on the websites for folks. That will also be available in the follow-up email. We did have a question saying, could leaving the rubber flap on the mower help distribute the clippings better?
Yes, actually, I like mulching mowers more than anything else. So mulching mowers normally do a fairly good job of getting the clippings back returned. I actually know of a number of homeowners that bag clippings. Sometimes you can use them for compost. I get a little worried, obviously, if they're putting a pre-emergent herbicide on and trying to compost those. I normally try to discourage bagging clippings. Most municipal city waste won't take those into their compost or their waste piles, their organic material piles. So I don't really recommend that. I like mulching it because you actually save on the fertilizer. The other aspect you can do is mow it twice. If you don't want to get off the mower, and you see that there's some clippings there, just go over it again. And particularly with a mulching mower, you'll take care of it.
Well, we are right at 12:31. So Dr. Miller, if you're going to stick around, we can maybe have time for some questions later. But now we will move over to Dr. Doug Richmond, turf entomologist, and he'll cover his bit. And again, feel free to throw questions in the chat box as we go through. I'll try to keep publications available to those as we go through them. But again, those should be available to everybody at the for the post, what do I want to say? The post hot topics email. So Dr. Richmond, you are up.
All right. Thanks, Jeff. Hi, everyone. I appreciate the invitation from Jeff to speak with you today. I appreciate the attendance. So I just want to give you a little bit of overview of my program and some of the things that I'll focus on some of the things that I think will be most useful to you. We're getting into that time of season where you're likely to get a lot of calls and people inquiring about white grubs in particular. So I'm going to focus on that for most of the presentation, some of the things that we're learning about these insects, especially in terms of management. But before I get into that, I want to talk a little bit about some of the research that we're doing. Some of it's quite basic research. And my graduate students and my postdoc are of course, the ones that drive the ship on that. I'm sort of the figurehead, but they're doing some really interesting work, and I wanted to share that with you quickly here. So let's see if I can get this thing to cooperate. All right. So before I get started, so I'm going to talk about turf insects, but specifically I'm going to focus on white grubs. I've got a bulletin available on white grubs and managing them. And if I put a QR code here, if you're quick on the draw and want to scan that QR code, it'll take you directly to that resource. But I at the end of the presentation, I'll also double back on this to my website where I have all of those resources available to you. They're also available on our Turfgrass, our Entomology extension website. All of the resources that have to do with turfgrass insects are there and easy to find. All right, so you know that these insects are a pretty common problem in turfgrass, and one of the things that they do is that they mine through the soil, eating plant roots. They ingest a lot of soil and organic matter in the process, and some of these insects, and particular Japanese beetle, is an invasive species. Right, we know it's spreading across the U.S. It got here in the U.S. about 1920, and has since been moving southward and eastward from Connecticut, where it was actually first introduced. But it's also become a worldwide pest. It's being distributed across the world, and its range is expanding. So we started looking at asking some basic questions about what is it doing to the soil, right? It spends 9 months of the year in the soil as a white grub. What's it doing down there? Are there any important changes it's causing in the soil, either biologically, physically, or chemically, that could have implications on a larger sort of macro scale? So my graduate student, Gordon McLeod, is looking at how these Japanese beetles in particular change carbon distribution in the soil and soil aggregate size distribution. So he's found some pretty interesting things. And when you look, soil is basically composed of a lot of different particles of a lot of different sizes, right? These particles, known as aggregates, are basically the functional unit of soil. And what he's seeing is that when these soils are infested with Japanese beetle, it changes the carbon, the amount of carbon in those aggregates across that size distribution. So no matter what size particle it is, there's more carbon in the soil. Probably, obviously, as a result of these insects feeding and mining through the soil. And of course, the excrement coming out the back end kind of lays the foundation for different soil aggregates to form. Not only that, but bulk soil in the carbon changes dramatically in the two largest size fractions. And the pre-existing organic matter and the inputs are greater in those size fractions. So basically what that means is that these things are sort of ecosystem engineers, right? They're changing carbon distribution and cycling in the soil, which could have implications on a global scale, right? Not only does that change the suitability of the soil for plants to grow in, it also changes, excuse me, fire truck in the background. It also changes the likelihood that microbes have access to that soil and can continue to cycle nitrogen and carbon through that ecosystem. My postdoc is working on the same project, but she has some interesting data that's come out looking at, of course, if that carbon distribution and that particle size is changing, that changes the availability of those sources to microbes. Also, in the gut of that insect, the Japanese beetle, it's basically acquiring all these microbes from the soil to basically help it degrade, consume, and turn all of that material it's ingesting into nutrients for itself. Out the back end, though, what we're seeing is these increases across the board in some important greenhouse gases from these soils that are infested with Japanese beetles. In particular, carbon dioxide, which is here in the top graph. You know, as larval density increases in the soil, you see an increase in carbon dioxide being emitted from that soil. The same with methane, which is a really important greenhouse gas. So more larval density, higher levels of methane being released from the soil. So what this kind of tells us is that as the climate is changing, the range of Japanese beetle is expanding, and it is actually sort of exacerbating those factors that are responsible for climate change by increasing greenhouse gas emissions from the soil. It's a really interesting story. It's a basic story, but it's something that nobody had looked at before, and we're really pretty interested in following this string where it leads us. Sorry, the other project I want to just mention briefly is a project by my master's student, Ryan Beard. We got a grant from the USGA, and we're partnering with a lot of different conservation organizations, the U.S. Fish and Wildlife Service, Pheasants Forever, and the National Wild Turkey Federation, and Burke Boilermaker Golf Complex, some native seed suppliers, Stantec and Martin Seed Company. And what we're trying to do is establish a roadmap for golf course superintendents who are interested in renovating their out-of-play areas into native prairie. And the reason we're doing this is because although golf course superintendents, some of them are adopting this practice, they're a bunch of just one-off attempts, right? There's never been data collected to give us a sense for when we should seed, how we should seed, how much preparation does it take to get it to an area that you're going to seed such that you have success, and what does that timeline for success look like? So we're doing this on the camping course at the golf complex here at Purdue. And Ryan is looking at two different factors. He's looking at the influence of seeding time, either a dormant seeding in the winter or a spring seeding, and seeding method, either broadcast seeding or using a seed drill. What are the effects of those variables on the establishment of the prairie community over time? The other side of that question then, and this is what it's looked like the first year, you can see, you know, there's a relative moderate amount of success in changing that plant community to something that looks more like a prairie. I was just out there this morning, and it's even more diverse. It takes time, right? But in hand with this, then he's looking at the ecosystem services that are provided by these plant communities, focusing specifically on pollinator communities. So he's also collecting a lot of bees out here, and we're looking at changes in the abundance and the diversity of bees that result from renovating these otherwise waste areas or out of play areas back into stands of native plants that you'd find in the prairie. So he's collected a lot of bees. This is, I think, one collection time that he has pinned here. He's looking at a lot of bees. Very interesting, a lot of diversity in these areas, a lot of rich species richness. So the bee community just, really takes advantage of all of these floral resources that are what we've put into those previously waste areas. So that's going to go on for one more year. And after that, we're trying to create a living laboratory out of this, give these, make these plant communities, these prairie plantings, basically a place for others who are interested in doing prairie research. We've got some people from FNR that are going to do some burns to burn those communities down, and add the fire cycle in. And at the same time, it's training people how to do these controlled burns. So we're trying to look for ways to leverage these efforts into to make them a resource for anybody who's interested in tagging on, right? We've now established them, they're coming along. So anybody has any ideas, I'm all ears. Let's, give me a holler, and let's talk about anything that you would want to do with these to take advantage of these plantings, whether it be just for demonstration, showing people what can result from these kinds of efforts, or if there are any other research projects that people are interested in working on. I am happy to partner with you on that.
Hey, Doug.
Yes.
Real quick. Well, one, I'm going out to an airport this afternoon, so I might do a name check for you. Because that would be something with those pollinators. But we did have a question going back to the white grubs. It was asked, what's the difference in damage that a white grub does to the soil compared to, let's say, an earthworm?
Yeah, so that's an interesting question, because really they do very similar things in terms of carbon and nitrogen distribution in the soil. What we're seeing is that these white grubs are doing some similar things to what invasive earthworms would be doing. So it's very similar. It's just that, of course, the white grubs also carry that extra impact of damaging the turf as well. Okay, I hope that answered the question. So let me talk about some of the more practical research we're doing with white grubs. I've enlisted one of our former students who was an entomology major and a graphic design minor. And we're starting to put out a lot of these infographics related to turf insects to help people understand their life cycles, what they're doing during different parts of the year, and basically when and how you can manage these insects. So this is one example. Most of you will recognize this as Japanese beetle, and this is available in that bulletin that I showed you at the beginning of the presentation. But we're trying to basically cover the waterfront on turfgrass insects by creating a bunch of these infographics that they're there for you to use. We use them in a million different ways. They're quite handy. They're quite beautiful, I think. But those are there for you as a resource if any of your clients are interested. So, of course, being a turfgrass entomologist, one of the things I'm most interested in is how grubs damage turfgrass. And we get a lot of questions about this, aside from what you might think is that they're feeding on the roots, right? Duh, that's a no-brainer. But I like to think of the way that grubs damage turfgrass in two separate ways. All right. First is this sort of primary damage that they cause that causes the grass to become wilty and then, of course, collapse, and it causes these patchy brown areas in the turf. This primary damage is a direct result of that that tunneling and mining through the soil that dislodges the roots, and the consumption of the roots that the grubs do themselves, of course, stops the plant from taking up water and nutrients. And that's when you see this collapse of the turf. But a lot of times what you see is no real apparent damage to the turf until the animals find the white grubs, right? The white grubs may not be causing any visual damage at all, but if the animals figure out that there's a nice patch of grubs there for them to consume, they go in and tear up the turf. I call this secondary damage, right? It's not caused by the white grubs themselves feeding on the plants, but simply their presence being attractive to other animals. Now, I want to see Jeff or if you could, anybody in the audience can guess the three most common animals we see associated with this kind of secondary damage.
Throw them in the chat box, folks.
Love to
if
see
you
what
can
you're thinking.
Throw them in the chat box. Ooh,
Yeah.
we got some rolling in.
Good. Let me see here.
Oh, Don, he's a good old master gardener. He's pretty good. Oh, we've got some good answers. You want me to read them off for you?
Yeah.
So first and foremost, we got raccoons. Is that one of them?
Yeah, that's a good one.
We've got moles. What about moles?
Moles, not this kind of damage. They will cause some damage when they're foraging for grubs, but it's not typically this bad. But yeah, it's a totally acceptable answer. Yes.
I've got skunks.
Yes.
Okay, this is one that I would have never thought of. Someone said wild hogs.
Absolutely.
That's new to me. I learned something today.
Yep. Absolutely.
And then we've got humans, armadillos, anything like that.
Yep. Armadillos will absolutely do this. I suppose humans could do this if they were interested in causing vandalism to turf. So what you've said there, I think, is we have a good list. Those two come to the top of the list. It's a lot of times it's skunks. That's the main culprit, but raccoons will do it, armadillos, wild hogs. Here's one that I think you will find surprising. This is actually more common than we think, and especially in rural areas. Turkeys, wild turkeys will actually find these patches of white grubs and shred the turf looking for them. So it's interesting, right, that sometimes we get surprised by the habits of animals and especially their foraging habits and what they will do when they find or locate a source of protein. So those are some good guesses. I appreciate that. Everybody seems to be on the same page here. So a lot of the more applied work that we do then is aimed at trying to figure out how to best manage these white grubs. So I work closely with industry to conduct a lot of trials. I've been doing this for close to 20 years now. So I've accumulated a lot of data on these different sort of insecticides, soil insecticides, when looking at things like application rate, application timing, when during the season is the best time to put these materials down to either prevent or control grubs once they actually are infesting the turf. So let me just share with you one of those sort of analyses that we've done. So I have here on the right the active ingredients in the insecticides, and I've got 5 of the most common ones: chlorantraniliprole, clothianidin, cyantraniloprole, imidacloprid, which is otherwise known as merit, and tetraniliprole. All of these are pretty common in the turf industry. And then I've got a number of trials, either looking at preventive applications, right? That's application that's made before the grubs are even present. An early curative application, that's an application that's made once the grubs are in the soil, but before damage occurs. And then a late curative application. That's an application that occurs later in the season when you're dealing with big, fat, robust white grubs and damage may be visible at that time. what you'll notice on each of these bars is an N equals, and that's the number of individual experimental trials that I've conducted with each of these different compounds at each of these different timings. So we don't have a lot with cyantroniloprol as a late curative product, only 2 replicated experiments, 3 with tetraniloprol in the late curative window. But some of these others, I have a lot of confidence in these data because it's not just a one-off application, one-off trial that, you know, you're going to have some variability. This is actually whole sets of entire experiments that I've accumulated over time. But what you'll notice here is that a lot of these products work pretty well in a preventive mode. You know, the difference when I say 70-some% control, that's still pretty good grub control, right? Yeah, it looks bad compared to something that's going to give 95% control. But if you can knock out 70% of the grubs in an infestation, you're not likely to ever see damage. You can see how that same sort of pattern holds true with the early curative. So a lot of lawn care operators will go on this preventive, will incorporate this preventive approach into their management regimes. That's mainly out of necessity. If you have 500 lawns that you have to visit and put a grub control down, maybe some other things, fertilizer at the same time, you're going to need a broad window. So those guys like to start in June and make those applications preventively. But what these data say is that you don't have to do that. You have a much longer window than just June. You can get into July, August, right? And you're still going to have a lot of success with any of these newer chemistries. Even, and this is the most surprising, even in this late curative window, when you're dealing with late instar white grubs, second and third instars, maybe even damages starting to become apparent, most of these newer chemistries do extremely well under those conditions. You can see some drop off with some of them. This is about, you know, we're going below 70% control here. So we're getting into that realm where, okay, maybe it's not the best approach with this compound, but for the most part, any of our modern white grub insecticides still do a pretty good job up until mid-September. That's what these late curative application timings are. Now, let me stop there for a second. Does anybody have any questions on this relative to grub control?
One question here, Doug. It's actually from me. How do you measure the threshold in terms of how many grubs is too many in terms of, you had mentioned secondary damage, but in terms of primary damage, if I have a lawn and I have grubs, but is there I know you guys do measurements, but how would I decide, hey, it's time for me to do this application or think about it? Because if I don't, I'm going to have some either primary or secondary damage in the very near future.
That's an excellent question. So, and I don't have a great straightforward answer with you. I'll try to make this as useful as possible. For the secondary damage, the damage caused by animals, we don't have really any good thresholds. They'll tear up turf, even if there are relatively few grubs in there once they find them. But that's not something that's very easy to predict. It really depends on the animal and how hungry they are at the time. So we don't have a good threshold for that. In terms of the primary damage, we go with a rule of thumb of about 10 white grubs per square foot as being sort of a threshold. But the most important factors that influence that threshold are going to be, what is the status of the turf to begin with? Is it a well-maintained lawn that gets plenty of water, is well fertilized? If that's the case, if it's a high-quality lawn, a well-maintained lawn, that threshold can go clear up to 20 grubs per square foot and beyond. If it's a poorly maintained lawn, that threshold can be as low as 5 per square foot. The other factor here is it depends on the grub species. Remember, it's not just Japanese beetle. Actually, Japanese beetles are pretty wimpy compared to some of our other white grub species. So where I would say a threshold on average for Japanese beetle might be 10 per square foot in the average lawn. For something like European chafer or Asiatic garden beetle, insects that we find in the northern parts of Indiana, that threshold can go down to 5 pretty quickly. So I'm sorry that there's not a real great straightforward answer to that, but it depends on status of the lawn, health status of the lawn, and species of white grub that you're dealing with.
Oh, that does help out, especially because we know golf courses are high maintenance, and then we have our home lawns that can be high maintenance, medium maintenance, or low maintenance. But no, that helps out tremendously. We've got 2 more questions. How early can you apply in the spring and still be effective? And then how far down in the ground do the grubs live?
Okay. So first question, how early in the spring can you make an application? So a preventive application, if you're using the right material, we've pushed it clear back into the first part of May, and you could still get effective grub control, even though the grubs don't show up until July. I don't recommend that approach simply because you're putting on an insecticide way early. You're exposing it to a lot of things like runoff, dilution with soil, with rainwater, microbial breakdown of that product, and you still don't know for sure that you're actually going to have a grub problem to begin with. But the short answer is you can push most of these products clear back into May. The second question then, how far do the grubs go, or where are they located in the soil? Let me back up just a couple slides here. Okay, so it depends on the time of year. During that time frame between July and in September, those grubs are pretty high in the soil profile, top couple of inches. All right. As the soil temperatures cool then in the fall, they'll start to move down in the soil profile. And when it's in the dead of winter, so assuming that the ground is going to freeze, which sometimes it does, sometimes it doesn't in this part of the country, those grubs have been found down as far as 12 to 15 inches in the soil. And they'll stay down there if it's cold all winter long, and then come back up toward the surface again to feed on the root zone as the soil temperatures warm in the spring. All right.
And then one more question here for you, Doug, as well.
Okay.
So in central Indiana, are the Japanese beetles the primary problem?
They are one of the primary problems. Japanese beetle and masked chafer. And that masked chafer is a complex of species that look very similar. So it's really those 2 species that we see most often. But yeah, Japanese beetle is right up there as the primary problem. Yeah, Japanese beetles and masked chafers. Great questions, you guys. Okay, let me just finish up here. So we've got some chemistries here that we can use against white grubs. And I like to think of them as, some of them are better for preventive, some of them are better for early, or are equally good as early curatives, and some are better for late curatives. But we also have some biological options I want to mention. And some folks are really interested in, not using chemicals on their lawns. And I get that. The only problem is that with our microbials, we only have a relatively few that are going to be dependable enough and perform consistently enough to be worth the money. The first one that I'll mention is this Bacillus thuringiensis gallerii. It's marketed as a product called Grub Gone. It's a granular product, and if that's put down in an early curative fashion, we can see pretty good grub control from that product. Of course, these biologicals are a little bit more expensive than most of the chemical options, but if that's something you're interested in, you do have options. You just have to spend a little more money. I want to point out this Paenobacillus papilii. otherwise known as milky spore. All right. This product, the commercial formulation, first of all, is only effective against Japanese beetle. It doesn't really touch the other white grub species. The second thing I will say about that product is you really have to put that stuff on pretty heavy for a number of years before you build up enough inoculum in the soil for it to have a detectable effect on the white grubs. You know, every time somebody tests this product as a one-off, it is difficult to see that it's having much effect at all. There's a lot of marketing behind this product, but if you're looking for an effective biological control, I would lean toward the Bt galeriae or these insect parasitic nematodes, one called Heterorhabditis bacteriophora. I know that's a mouthful, but these are available from commercial suppliers, and they are quite effective. As long as you put them on as an early curative, they do well. And the key thing is you can't spray these things on the lawn and expect them to sit in the canopy of the turf and bake in the sun and have any survival rate. You have to put these things on either in the evening or early in the morning when the sun isn't directly overhead, and you have to water them in, get them off the leaf blades and into the soil where they have a chance of foraging and finding the white grubs to infect. Now, I want to wrap this up real quick, but here's what an infected white grub looks like. You see that pink color? That's a result of that grub being killed by these insect parasitic nematodes. Those things actually penetrate the cuticle of the insect. They get inside and they release a bacteria that actually kills the insect. And then that bacteria turns the inside of the insect into a slurry. And that slurry then serves as a food source for the nematodes to reproduce, produce the next infective generation of infected juveniles to go and infect other insects. So last thing here, here's my contact information. I have these resources available to you. Reach out to me if you have trouble finding them, or if you have any, need any help with sorting a turfgrass insect problem. I always make myself available to you. I'm happy to speak with you. So please keep that in mind and don't be shy about contacting me.
Thank you, Dr. Richmond, and thank you, Dr. Miller. I'm going to get on my turf soapbox here and just say, we get a lot of comments. Oh, it's just turf. You just have to mow your grass. There's so many there's so much research that's done at the campus level for different levels of maintenance. So I appreciate both our pathologists and entomologists for speaking with us today. Feel free to stay on for a bit. There were a couple other questions. For those of you who are still on, there is a link for the post-program survey. So if you don't mind answering a few questions there. We did have a couple questions right now. I've got one for Dr. Miller and one for Dr. Richmond. Let's say my county extension educator, Jeff, who's more of a weed guy than a weed sciences guy than a pathology guy. How would I go about sending a turf sample that might have a disease to the Purdue Pest and Diagnostic Lab?
Thanks, Jeff.
Good. He put it right in there, so we're good there.
Yep. So that is the link that you'll follow. We require pictures. So we're going to go and throw that out there. If you're not going to meet us halfway and at least use a phone and request it from the client or get it while you're out for the site visit, us having the stand symptoms is extremely important. Now, that being said, we often can't diagnose just based off the stand symptoms. We often have to have that physical sample in hand. But at least we're able to put those pieces of the puzzle together. There is a form there that you'll fill out. I believe it's $11 for homeowners. We haven't raised that at all. We've gone a little bit higher for golf courses and for professional. But I think that's a pretty good deal. We do get a lot more homeowner samples and landscape samples than I ever received in Missouri. I think that's just kind of a testament to how much more organized the lawn care associations are here. But You also can, so the place to submit the pictures is actually, I'll put this email, it's just turf-samples@purdue.edu. And that goes to everybody. So that goes to John Bonkowski, to Tom Creswell. We've really centralized that. And I really appreciate those guys for helping me when I have to do this, because I know I'm coming home to some samples. They can at least start that processing process for me before I get back. So that way I can hopefully streamline and get back to everybody as soon as I get back.
There was a question about can you safely use BT in a garden plot? That'd be for our entomologists.
Yeah, the answer is absolutely yes. You know, it's one of the safest products out there, has essentially zero toxicity to humans or any other vertebrates. So yeah, I wouldn't hesitate at all.
All right. Well, again, we really appreciate you guys coming on and sharing the research that you do and the questions you answer for our turf-related programs. Everyone who's on the call, you know, we appreciate you guys being here today. And with that, a lot of information, and we appreciate you guys being on, and everyone have a good rest of your day.
All right. Thanks, everybody.
Yeah,
Have a good
thanks,
one.
everyone.